This is part 2 of the Chapter 12 lecture, and we left off in part 1 talking about the S or synthesis phase of the cell cycle. So we were talking about how many errors there are, and there's only about one error per 1 million bases of DNA that gets replicated. And with 3 billion base pairs in the mammalian genome, the average one, that means there's only 30 errors per cell cycle. These errors are mutations that are occurring in the somatic body cells. So they actually do provide some mutation, but it's a very, very low rate. So again, the reliability of replication is pretty amazing. So after DNA is replicated, that chromatin, which is again the DNA and protein complex, condenses. And it does that by coiling and folding to make a very tight, small package. And then before DNA replication can occur, that is actually going to have to uncondense or loosen up in order to get our good DNA copying. This is a picture showing the actual replicated DNA. So we have this double-stranded mitotic human chromosomes, and this is usually what we think of when we think of chromosomes. But really, one chromosome is just one long strip of DNA, and it's only after replication that we get this two set of chromatids in this classic shape that we see here. But before we go on, I have another joke for you. How do you eat DNA spaghetti? With a replication fork, of course. Haha. Okay, so the chromosome. So each duplicated chromosome consists of two sister chromatids, which are joined together, and each of those chromatids is an identical copy of the chromosome's DNA. Initially, when they're first replicated, those chromatids are attached all along the length of the strands, and they're held together by protein complexes called cohesins. I have that over here in the pink. So the cohesins make this tight structure that you see right here. But as time goes by and as the chromosome condenses more, the region where the chromatids connect shrinks to a very narrow area that's made up of two centromeres. And so we can see that here. These two centromeres actually become the only place where the two chromatids are attached. These centromeres are actually a specialized region on the chromatid that have specific DNA sequences. Usually they're long series of tandem repeats, sometimes hundreds of thousands of bases long. So that's a lot of DNA, but they have yet to really sequence all the DNA that is in that centromere section. And so there is still a lot to learn about what actually is the usefulness of that tandem sequencing of DNA that's located there. So it's not just proteins or something, it's actually DNA that is forming that centromere. What happens in the mitotic process is that proteins bind to these centromeric DNA sequences, and they attach the two chromatids to each other at that region. And this long area that's on either side of the centromere is called the arm of the chromatid. Okay, so the sister chromatids eventually are going to separate and move into two new nuclei at the opposite ends of the parent cell so that each daughter cell is going to have an exact copy of the DNA. So when we're looking at this structure right here, we're looking at two sister chromatids that are representing one chromosome. So together they make up a chromosome. But once they separate, they're actually a set of chromatids. And this is really vocabulary that I just want you to be familiar with. Because it's not until they actually separate that they're actually called chromosomes again. So until they separate, they're really considered sister chromatids. And when they separate, this single-stranded chromosome is what we term a chromosome. Thinking back to your biology days, the two sets of sister chromatids that are the same sequencing of DNA are the homologous chromosomes. And the homologous chromosomes, remembering back, one is from the mother and one is from the father. So whether we're looking at them as one strand or as double-stranded, we have a homologous set when we look at one from mom and one from dad. Okay, next slide. I'm not sure this is going to work. We'll give it a try. Well, nope, that video was not going to work. So you can find lots of videos online on what the steps of mitosis involve. But for now, we're just going to talk through them. So the first step of mitosis is prophase. And the key characteristics of prophase are that the nucleus disappears and the nuclear membrane breaks down. So you're going to need to have these key events memorized for each of the steps of mitosis. So make sure you get that clear if it's not yet. But what's happening essentially is the mitotic spindle is beginning to form. The chromatin is condensing. We have visible chromosomes. The centrioles are moving to opposite sides of the cell, what we call the poles. Of course, that's only occurring in animal cells. Plant cells do not have centrioles. Protein fibers are crossing the cell to form the mitotic spindle. The mitotic spindle is made up of microtubules, primarily made out of proteins actin and myosin. And the mitotic spindle is very important in coordinating the movement of the chromosomes to each side of the cell. In animal cells, the centrosome has a pair of centrioles at the center, but the centrioles themselves are not essential for cell division because what they've found that if they're missing in an animal cell, like if they artificially destroy them by laser, for example, they still can have a spindle form during mitosis. And the centrosomes of most plants lack centrioles, but yet plants still form mitotic spindles. So it's really a little bit unclear what role or how essential they actually are. But basically, as mitosis starts, the two centrosomes are located near the nucleus to begin with. But as the spindle microtubules grow from them, the centrioles start to get pushed apart by the growing microtubules. What is called an aster is a radial array, like a spoke, of short microtubules that extend out from each centrosome. So that aster actually is what's pushing the different centrioles apart. So what we end up with in the spindle is the centrosomes, the spindle, microtubules, and the asters. Now we have a lot of C words here. We have chromosomes, we have centrioles, we have centrosomes, we have chromatin. I would really recommend making a list of all of these and draw pictures or just something to help you keep them all straight, because frankly it's a little bit confusing because they sound very similar. So now we go into prometaphase, and the key characteristics of this next phase is that the chromosomes begin to move. The spindle is actually becoming more set, and the spindle fibers are attaching to the centromeres, creating what we call kinetochores. And the microtubules are attaching to these kinetochores, connecting centromeres to the centrioles. So let me just go through this in a little bit different way because I know this sounds very confusing. Each sister chromatid of a duplicated chromosome has a kinetochore. That's made up of proteins and chromosomal DNA. The kinetochore of the joined sister chromatids actually face in different directions. So you can see here we have the kinetochores attached here at the center of these sister chromatids. When a chromosome's kinetochore is captured basically by microtubules, that is actually what's happening during prometaphase. So some spindle microtubules, they actually have a special name called kinetochore microtubules, attached to these kinetochores of the chromosome here. So now the chromosomes, as they're attached to these microtubules at the kinetochore, that attachment actually begins the pulling apart of the sister chromatids because the chromosomes will begin to be pulled towards the two centrioles at the poles. The microtubules, as we've mentioned before, of the spindle are attached to each pole. And so when they're attached to the pole, that there is an actual tug-of-war that is going on with the chromosome, the sister chromatids. So we have one set here going off and attaching to one spindle pole. Then we have this one going off and attaching to this spindle pole. And that tug-of-war that actually occurs now, there is pressure put on this set of sister chromatids so that they're being pulled at from both directions. And that pulling motion just happens to be the cause of what lines up the chromosomes in the middle of the cell, or what we call at the metaphase plate. So this is when the chromosomes are going to settle here, midway between the two poles of the cell. And it's because of the action of the microtubules in the spindle attaching to the kinetochores. You can see in this picture that not all of the spindle fibers attach to the chromosomes or the sister chromatids. So those are called non-kinetochore polar microtubules, and they are believed to actually still help guide the sister chromatids into the correct pole or correct side of the cell division. Once this process has occurred, once we have the attachment of the sister chromatids to the spindle and the spindle fibers attached to the poles, we say that the spindle is now complete. So now that we have our chromosomes lining up very nicely across the metaphase plate. Meta means middle. And again, it's happening because of those spindle fibers coordinating the movement and balancing the pull in both directions. That tension of having these chromosomes line up this way is really awesome to ensure that the chromosomes are going to separate properly so that each new nucleus is going to get only one copy of the chromosome. Here's another picture showing that. This is an actual photograph showing the microtubules there attached. Right there we have the centrosome attached here with the kinetochores. You can also see it in this picture here. This is with an electron microscope. So once we have metaphase finished, we have those chromosomes lined up, we move into anaphase. So I always think of anaphase starts with A. This is the portion where The chromatids will be pulled apart,